Okay, so what if I told you that we can take one of your skin cells and just rewind its biological clock? Turn it all the way back to the beginning, to a state where it could become literally any other cell in your body. Now, it sounds like pure science fiction, right? But it's very real. And for years, it was a process that had about a 1 in 100 chance of actually working. That is, until one incredible discovery changed absolutely everything. Let's get into it.
So to really wrap our heads around this breakthrough, we've got to start with a super fundamental question.
You know, we've always dreamed of turning back time, right? Well, that's still in the realm of movies. But what if we could do it on a much, much smaller scale, like for the actual building blocks of our bodies?
Well, for our cells, the answer is a massive Nobel Prize-winning yes. Seriously, back in 2006, a scientist named Shinya Yamanaka figured out that with just 4 specific genetic factors, he could basically force a mature cell, think of skin cell, to completely rewind its own history.
And what this process creates are these things called induced pluripotent stem cells, or iPSCs for short. The easiest way to think about them is like a biological blank slate. You can take a skin cell, hit the reset button to turn it into an iPSC, and then you can tell that iPSC to become a new heart cell or a neuron, pretty much anything you want. I mean, the potential for medicine here is just staggering. But, and this is a huge but, there was a major catch.
So yeah, this discovery was a total game changer, absolutely revolutionary. But then, when other scientists tried to actually do it, they slammed right into a brick wall. The reality in the lab was, well, it was super frustrating because most of the time, the process just failed.
I mean, the success rate was shockingly ridiculously low. We're talking about 1%. 1. So for every 100 cells you tried to reprogram, on average, only a single one would actually make it all the way back to being a stem cell. Just one.
So you gotta ask, right? What happened to the other 99? Well, most of them just got stuck. They get trapped somewhere in this weird in-between state. Not quite their old self, not quite a new stem cell, and they'd eventually just give up. For years, this massive inefficiency was the biggest roadblock in the entire field.
Okay, so to get why this is so tough, scientists have this really cool analogy. It's called Waddington's epigenetic landscape. Picture this. A mature cell, like your skin cell, is a ball resting at the bottom of a deep valley. It's stable, it's happy there. Reprogramming is like trying to push that ball all the way back up a super steep mountain, fighting against gravity the whole way. You're literally battling against all of the cell's natural programming, all these chemical marks that are screaming, you are a skin cell, stay a skin cell. It's a massive uphill battle against the cell's very nature.
So the big question was, why was it so rare? And the scientific community was basically split into 2 camps. One idea, called the stochastic model, was that it was all just random luck. A cell had to get this perfect, statistically improbable sequence of events to make it. The other idea, the elite model, was that maybe only a tiny handful of special or elite cells were even capable of changing in the first place. Well, turns out, everybody was wrong.
You see, the real answer was so much cooler. It had nothing to do with luck or special cells. It was all about an active internal fight.
And this right here was the total paradigm shift. That low success rate, it wasn't a bug in the system, it was a feature. The cells have a built-in safety mechanism, like an emergency brake, specifically designed to stop them from just randomly changing their identity. And scientists finally found the molecular switch for that brake.
Okay, so meet the main character of our story, a protein called MBD3. It's a key part of this huge molecular machine called the NuRD complex. And you can think of this NuRD complex as the cell's high-tech security system. Its whole job is to find all the genes that say, be a stem cell, and then lock them down tight by packing up the DNA around them so they can't be turned on. And MBD3, it's basically the master key that controls that whole lockdown.
And this is where things get really, really wild. Scientists figured out that those Yamanaka factors, the ingredients that are supposed to cause the reprogramming, were doing 2 totally opposite things at once. On one hand, they're hitting the gas, trying to switch on the stem cell genes, but at the exact same time, they're calling in the MBD3 protein to slam on the brakes and shut those very same genes right back down. It's this insane, constant tug-of-war inside every single cell. So the reason only 1% succeeded is because, by pure chance, they somehow managed to win that internal fight.
So once the scientists finally understood this whole gas and brakes paradox, the solution became almost beautifully simple. This team of researchers basically said, Okay, wait a second. If MB3 is the emergency brake that's actively stopping this whole thing, what would happen if we just cut the brake lines?
And this idea completely flipped the old model on its head. Suddenly, reprogramming wasn't this random, stochastic game of chance anymore. If you remove the brake, it becomes deterministic. It's predictable. It's like a factory assembly line. You take away the resistance, and the cell just reprograms every time.
And the results? Oh, they were absolutely breathtaking. When they got rid of the MBD3 protein in the cells, the success rate didn't just inch up a bit. No, it jumped from that measly 1% all the way to nearly 100%. 100%. The whole thing went from being a month-long lottery ticket to a totally predictable, week-long process.
And just look at the speed difference. What used to take 3, even 4 weeks of just hoping and waiting now happened all at once, with almost all the cells converting together in about 7 or 8 days. They hadn't just found the emergency brake. They had figured out how to cut the lines and put the pedal to the metal. Okay, so this brings up a really obvious question, right? If we've got a method that's way faster and basically has a perfect success rate, why isn't this the standard for everything?
And this is where we hit that critical point where lab research meets the real world of medicine. And the answer really boils down to a simple trade-off. Which do you want? More cells or the perfect cell?
See, in a research lab, getting to 100% efficiency is like finding the Holy Grail. It lets you study the process perfectly. But remember, to do it, you have to genetically mess with the cell, right? You have to remove a key protein. Now, when you're talking about making cells to put into a human being, the number 1, 2, and 3 priorities are safety and perfection. You have to be absolutely sure that cell is flawless and won't cause any problems later on. So for a patient, one single, perfect stem cell is way, way more valuable than 1,000 pretty good ones.
So that's the key takeaway here. The MBD3 discovery was so monumental, not because it gave us a new therapy right away, but because it cracked the code. It proved that a cell's identity isn't just a passive thing. It's an active program, a program that fiercely protects itself, but a program that, if you know what you're doing, can be rewritten.
And unlocking that fundamental mechanism, well, it's opened up a whole universe of new research. Finding the cell's emergency brake wasn't really the end of the story. It was the key that unlocked a door to a whole new level of understanding. And it leaves you wondering, right? What other cellular programs are just waiting for us to figure out how to rewrite them?